Hi everyone, I'm going to explain the ELISA experiment in this video, and I really wish you guys were doing this experiment in lab because this is one of the experiments that's being done for detecting coronavirus infections right now. So there's multiple tests that people are coming up with and labs are coming up with to detect if people have the novel coronavirus, if they're infected with it, and ELISA is one of those tests. So we're going to talk about what the ELISA is, what the lab is, and then in the end I'll talk to you specifically about the test that's being used with COVID-19 infections. So here on this picture, we see this micro titer plate. I'm going to describe what that is, but this is what you would, if you were actually doing the lab in lab and we weren't doing a quarantine, this is what you would have. It's a plate that has these little plastic wells, and this is where you run the ELISA experiment. And then here in this image, which we're going to learn about, you're going to see what we're trying to detect. So in these wells, we're trying to get antibody and antigens to bind to each other, and when they do, you get a color change, and you could use it to detect different things. So we're going to talk about the background of that. So ELISA stands for Enzyme-Linked Immunosorbent Assay, and it's an immunodiagnostic test that's used in lab to detect specific antigens or antibodies from an unknown sample. And just as a reminder, antibodies, we find them in secretions, we find them in blood. So you can use ELISA to detect the presence or levels of different chemicals, whatever you're interested in. So let's start here. So you can use ELISA to detect if drugs are present in someone, hormones, foods, pathogens. And the ELISA lets you can do various ELISA tests. You can do it to just detect if something is present or not. So you can look for a pregnancy hormone, or you can look at specific levels of different things that are important to you. So here, foods are used as allergens. So you're seeing if someone's making an antibody to a specific food. So it's a very useful test that's used in labs all over the world. And you typically run the ELISA experiment or the ELISA test in a small plastic well called a micro titer plate. So it's this plate and it has 96 wells. Wells are these little openings. So this is the micro titer plate. And usually you use one with 96 wells. And we give all the students 4 rows just to save material. This is what it looks like, and I will discuss the experiment, but when you get antibody and antigen binding to each other, you get a color change. So this is what we're seeing in this plate. And this whole experiment, all of ELISA, is based on antibodies attaching to their antigens. And when an antibody attaches to its specific antigen, you get an antigen-antibody complex. So for example, if you've had the flu before, if you've had the influenza virus, you've had a specific strain, you probably make the antibody for that influenza virus so that if you're ever exposed to that virus again, your antibodies will attach to the virus and they will get rid of it in one way or the other. So we're going to talk about it during the immune system lecture. So this is how antibodies and antigens work. And in the case of the coronavirus infection, so just to make this relevant to you guys, the antigen is actually the coronavirus, the SARS-CoV-2 virus, which is the virus that causes COVID-19, the disease. So that would be the antigen, and hopefully your body would make antibodies to attack the coronavirus, and then you would have those antibodies. So that's the ELISA experiment, and you could do ELISA for many different things. So it's all based on antigen-antibody specificity, and antibodies are very specific to the antigens they bind to. So an influenza antibody will only bind to influenza, the virus. So you can use ELISA to detect unknown antigens or unknown antibodies. And in both tests, whether you're trying to detect if someone has an unknown antigen, like the pathogen itself, or an unknown antibody in their blood, in both tests, I'll describe it, you use something called an antibody enzyme conjugate, which is added to the antibody antigen binding to each other. And this will make more sense in a second. And this antibody enzyme conjugate will catalyze a color change whenever it sees its substrate, and its substrate in this experiment is chromogen. So every time the antibody enzyme conjugate sees chromogen, it'll do a reaction, and then you'll get a yellow color change for this experiment, for purposes of this in class. So actually, let me move myself so this makes more. Okay. So there's two types of ELISA. Well, there's qualitative ELISA and quantitative ELISA. Qualitative ELISA means you just care if something is present or not. So it's just saying, are you pregnant? Yes or no. You don't care how pregnant someone else is. So that's a quantitative ELISA. That's a qualitative ELISA. And then a quantitative ELISA is when you're actually trying to quantify something. So you're trying to look for the amount or level of substance measured. So maybe if you were doing a drug test on someone, you actually wanted to know the amount of a specific drug, you can do a quantitative ELISA. And in this lab, had you guys actually been doing the lab, you are doing a quantitative ELISA. So you're trying to detect the amount of an antibody, and we'll talk about it in a second. And then for quantitative ELISA, the way you tell how much someone has something, you read the color change, how much the color changed in a machine called a spectrophotometer, and it gives absorbance readings that tells you how much antibody or antigen they have. So again, this is for the quantitative ELISA, not for the qualitative one. And high absorbance means you have a high amount of antigen or antibody, whatever it is you're looking for. And you compare the results to known amounts of the antibody or antigen you're testing, and we call those standards. And then you plot them on a standard curve. So with the ELISA, there is direct ELISA and indirect ELISA. And we're going to start off by talking about the direct ELISA experiment. So in this image here, you have a well. We put everything and then discuss it. Okay, so this is a direct ELISA, and it tests for an unknown antigen. So what you do is imagine your plate that I said has 96 little wells. If we look at one little plastic well, it has a known antibody stuck to the plastic. No, go back. Okay, it has a known antibody stuck to the plastic. And what you do is you add the sample. Let's say you're trying to figure out if someone has an unknown antigen. So does someone, maybe you have the antibody, actually I'll talk about it in a second. So you have an antibody that's known, you add their sample, you're trying to see, do they have drugs in their blood or something like that. So you add the unknown antigen, and then you wash away anything that didn't bind, because the antibody and the antigen will bind to each other only if they're specific for each other. Then you add your antibody enzyme conjugate, and then you add your substrate. And every time this whole sandwich sees the substrate, it'll lead to a color change. And the substrate is chromogen. And a color change means a positive test. Yes, the antigen is present. No color change means it's not, because the enzyme antibody conjugate did not catalyze a color change. And the direct ELISA is sometimes called a sandwich assay, because you have this nice symmetry between antibody, antigen, antibody. And with the direct ELISA, an example of a direct ELISA is a pregnancy test. So with the pregnancy test, we use an antibody that specifically detects the HCG hormone. That's one of the pregnancy tests. So every time the antibody binds to HCG hormone, it causes a color change. And that's the same thing as when you think of a typical pregnancy test. When you see the strip come up, it's because that reaction happened. It's a very quick reaction. Another area where we use a direct ELISA is, there's different influenza tests, but we can do an influenza test where we use a known antibody, and then you add a sample from someone, and if they have the antigen, specifically that influenza virus, and it binds to it, you see a color change. So this is a direct ELISA, and you're detecting an unknown antigen using a known antibody. In indirect ELISA, it's the opposite. You have a known antigen and you're trying to detect unknown antibodies. So we use indirect ELISA, for example, with HIV screening and other things, and we're trying to see if someone has HIV antibodies in their serum or their blood. Serum is a component of blood. So what you do is you take a blood sample from a patient and then you put it on the wells, and the wells are coated with HIV antigens, and if they have the antibodies, they will bind. So that's an example of an indirect ELISA, and it's preferred because HIV antigens often hide in infected cells, so we wouldn't want to do a direct ELISA, because with a direct ELISA, you have the antibodies here. So it's the opposite, and you look for the antigens. But with HIV, a lot of times you cannot detect the HIV antigens because they're in infected cells, and it would be too hard to find them in blood samples. So again, this here was the indirect ELISA. The one before was the direct ELISA. So with an indirect ELISA, you have a known antigen and you're testing for an unknown antibody. With a direct ELISA, to go back to that one, you have a known antibody and you're trying to look for an unknown antigen. And with both of them, direct and indirect, you have this antibody enzyme conjugate, and you add a substrate to it, and if you get a color change, it means you have the presence of whatever it is you're looking for. Whether you're looking for to see if someone's pregnant, or they have HIV, or they have coronavirus, which we'll talk about later. Okay, so in lab, you're specifically, what you would have been doing, unfortunately you're not, is you were going to look for IgA antibody. So let me put all of this out. So in this lab, you are going to do a direct quantitative ELISA. Direct meaning it starts out with wells that are coated with antibodies, so known antibodies. Quantitative means you're going to quantify, to put a number for whatever you're looking for. ELISA, to test for salivary IgA antibodies. So IgA is an antibody. There's different types of antibodies. We're going to learn about them in class, like IgG, IgM. Anyway, IgA is an antibody that's found in various secretions of the body. So it's found in saliva, it's found in along your respiratory tract and digestive tract. So like the nasal membranes, you see mucus, and in that mucus you have IgA. It's found in tears and it's found in breast milk. And what IgA does is it protects you from pathogens. So that's what antibodies do. They protect you from pathogens. And this is the antibody that we see in secretion. Specifically, we see it in saliva. This is what you're looking for. So we see it in all these other things. This is why breast milk is very healthy, because it has antibodies that will protect the baby against many diseases that they could potentially be exposed to in the early weeks of life. And this is also why your eyes are not always infected, because tears protect you against so many pathogens that your eyes are being exposed to. So you're going to look for how much IgA do you have in your saliva, and lower levels of IgA can contribute to potentially more infections and allergies, because again, IgA is an antibody that protects you. And so you were supposed to look to see how much IgA do you have in your saliva. And IgA, what it does is it can protect you against many pathogens, including bacteria that cause cavities. So you are all going to see your amount of IgA using an ELISA experiment. So you were going to do a direct quantitative ELISA to test for IgA antibodies. You're testing how much IgA do you have in your saliva, and this is important because it could potentially tell you how healthy you are. And IgA levels vary in the time of day, how young you are, how old you are, how healthy you are, genetics, a lot of things vary. That's why, for example, some people who never get a cavity and some people who always get cavities. It can be contributed to a lot of things, but one of the things it can be contributed to is how much IgA antibody do you have in your saliva. And here we're gonna have the IgA antibodies act as antigens because we're doing a direct ELISA. So the wells are going to come with antibodies that specifically bind IgA. So we're going to pretend that IgA is the antigen. So this is why we're going to call this a direct ELISA. So you have antibodies that will bind to IgA, which is also an antibody, but we're going to pretend it's an antigen here for the antibodies on the wells. And then you're going to add your antibody enzyme conjugate. Then you'll add the substrate, which is the chromogen. And every time you see a color change, it means that the IgA antibody in saliva is present. And then you're going to quantify how much did the color change to figure out how much IgA you have in your saliva. So for this direct ELISA, the test wells have Go anti-human IgA antibody, and this binds human IgA antibody. Okay, so this is what you would be doing in lab. I'm so sad that we're not actually doing this experiment, but I'm going to try to explain it as best as I can. So you have this microtiter plate or these test wells. So every little circle here is a test well, and they have anti-human IgA antibody adhered to them. So you can't actually see it, but if you were to be able to imagine it on the well, on this little circle, you have anti-human IgA antibody, which will bind every time it sees IgA antibody. So what you're going to do is you're going to add your saliva sample. You're going to dilute it first. You're going to take your saliva sample, you're going to dilute it with water, and then you're going to pipette it on the well. And remember that the IgA from your saliva will bind to the antibody that's stuck on the saliva that binds IgA. Then you're going to wash the wells because you want to wash anything that didn't bind. So your IgA here, here's the IgA, it will bind to the anti-human IgA antibody on the wells. You wash it, you basically add a chemical, it's mostly water, then you dump out the microtiter plate, then you add more of the wash, then you dump it out again, then you add more of the wash and you dump it out. And hopefully you've done good washes where only the IgA from your saliva is bound to the antibodies that are stuck on the wells. So the next thing you want to add is the antibody enzyme conjugate, and the specific enzyme it has on it is horseradish peroxidase or HRP enzyme. So that's the next thing you're going to add. And this antibody enzyme conjugate, which has the HRP, will bind your antibody antigen sandwich and create a complex. So then you're going to do more washes. You're going to let it sit, let them bind. Then you're going to do more washes, meaning you pipette some of the wash solution, which is mostly water, dump it out from the from the microtiter plate, pipette again the wash, dump it out again. And then you need to add the chromogen that the HRP enzyme acts on, and it's specifically TMB chromogen, and the TMB chromogen will catalyze a reaction where you have a color change every time it sees this HRP antibody complex. So you get a color change, and then for purposes of this experiment, you get yellow. It's not always yellow. It depends on what chemicals you're using, but if you were doing it in lab, you get yellow. So if we were to imagine that these swells all had our reaction, these ones that are yellow here all have IgA antibody, these ones do not. So then the idea is, well, let's say this was one person, this was another person, this was another person. How much IgA do they have? So how yellow is this compared to this? And this is where we're going to use a spectrophotometer. So a spectrophotometer determines how much absorbance is happening in there. So how much color, how strong the color is to make it easier. And then you're going to create a standard curve. Wait, no, let me go back. Okay, then you're going to create a standard curve to figure out what that absorbance means in regards to IgA levels. So I didn't tell you, but here on the wells, you have different amounts of known IgA levels that you're going to put on your well. Then you're going to put your unknown, your actual sample, and then you compare them with each other. And I know it's so much harder to understand without actually doing this in lab, but I'm really trying my best to explain it. So you create a standard curve. So after you put your plate in this machine called a spectrophotometer, the spectrophotometer will give you numbers and it'll tell you, well, A has this much absorbance, well, B has this much absorbance. And absorbance is basically how much color it's reading in the spectrophotometer. So then what you do is you take your standards. So on there you have wells that have standards amount of IgA. You're going to take your standards and your absorbance readings you got from them, and you're going to create a standard curve where the y-axis has the absorbance reading that you got from the spectrophotometer, or scientists sometimes we just say spec. And then on the x-axis, it's the IgA concentrations. You create a graph. And then what you're going to do is take your actual absorbance reading that you got from the spec. So maybe the spec tells you that your absorbance reading, I don't know, is 1.5. So then you go to 1.5 here, you go all the way across, and then you go down and you get your IgA reading. So you get your absorbance readings from the spectrophotometer. The x-axis is the concentration of IgA standards. The y-axis is the absorbance readings. You draw a best fit line, and then you get your own saliva sample, and then you do some calculations, which I'm gonna skip because you guys unfortunately are not actually doing this experiment. So the objectives, what I want you to know for your lab practical is, I want you to know that what did you actually specifically test for in this lab. And I'm really sorry that you guys did not do this, but you specifically tested for IgA in saliva, and what it signifies for your health is usually people with less IgA are more prone to infections, more prone to cavities. Know the components of the sandwich, what you created. Can I move this? Okay. What you created in your well, you had wells that were already coated with goat anti-human IgA. Then you added your saliva sample, which acted as the antigen, the human IgA. Then you added your antibody enzyme conjugate, and the enzyme was horseradish peroxidase. Then you added the substrate, which is the chromogen. So chromogen is the substrate, and every time this enzyme sees the substrate, it cleaves it, it catalyzes a color change, and then you get a yellow color. What's the purpose of creating a standard curve? The purpose of creating a standard curve is to count the number of IgA you have, to know how much IgA you have. And then given a standard curve and absorbance reading from an unknown sample, determine the IgA. So if you look at that curve that we get, or you look at that graph, can you figure out, if someone gives you absorbance on the y-axis, can you use it to figure out the IgA levels? Now to the somewhat fun stuff. So you were supposed to do an ELISA in lab and look at how much IgA you have. We're using ELISA, we've been using ELISA testing kits in science for so long, but one area it's been very popular is using them for COVID-19, so for coronavirus. So two test kits that I read about, one looks for IgG and one looks for IgM. So these test kits are taking blood samples from someone and they're seeing, do they have IgG or IgM? IgM is the antibody that your body makes in the beginning of an infection. So in the beginning, when you have the coronavirus infection, and then IgG you make in the beginning, but it stays long term. So hopefully if you've been infected and you've overcome the infection, you're relatively healthy, you still have IgG antibodies. So if you're ever exposed to the virus again, your body can fight it off. Although there's varying research about that. So they're ELISA kits. So here in this ELISA kit, if you look at it, they have their antibody, and then you could add controls or a sample from someone. So then that sample binds the antibody, and that sample is you use different parts of the virus to come up with the ELISA kit. Then you add your enzyme antibody conjugate, then you add your substrate, and then you get a color change, and then you read the absorbance using a spectrophotometer. So I hope that you guys learned something without doing the experiment and that it's relevant to what's going on right now.